Hi, welcome to my poster. This is Yuzhou Chang from Ohio State University. It is my great honor to introduce recent work collaborating with researchers from University of Missouri. Our work is to identify tissue architecture based on spatial resolved transcriptomics data. Tissue architecture is biological foundations of the spatial heterogeneity within complex organs like human brain, and thereby it is essential for us to understand the underlying pathogenesis of human diseases such as cancer or Alzheimer's disease. However, the current challenge of identifying tissue architecture is how to fully take advantages of spatial information. Therefore, we developed a novel deep learning framework, RESEPT, for characterizing, visualizing, and interpreting biological tissue architecture from spatial transcriptomics data via reconstructing and segmenting a transcriptome mapped RGB image. The RESEPT framework is starting from a gene expression or R velocity matrix. Empowered by spatial constrained graph neural network, the gene expression or R velocity matrix with spatial relations can be embedded into three-dimensional representations. Then the 3D embeddings are linearly rescaled to an RGB color spectrum and reconstruct a pseudo RGB for visualizing the spatial architecture. And finally, a pre-trained segmentation model based on convolutional neural network is trained to partition potential function region and obtain tissue architectures. The benchmark results are showed at Figure 2. We used the 16 dataset and performed jackknife cross-validation training strategy to train segmentation model. The performance evaluated by ARI was indicated that our tools is outperformed other 6 existing tools. To test stability of RESEPT, we use 2 high-quality datasets to simulate a series dataset with a gradual decreasing sequencing depth, and the result indicates the RESEPT was stable on simulating dataset. Notably, we also tried R velocity embedding on reconstructing pseudo RGB image, and we observed velocity can recognize a detailed sub-architecture that gene expression cannot detect it. To demonstrate the usage of RESEPT, we use human Alzheimer's disease generated from Department of Neuroscience to identify the tissue architecture and functional region. By inputting predefined markers, for example, layer 2 or layer 3 associated with AD development, RESEPT can reconstruct the image, a pseudo-color image based on selected marker, and enhance the image by filter out unrelated colors. Here we display the 3 RGB image reconstructed by layer marker, cell type marker, and A beta region associated genes. So the results indicate RESEPT can recognize important tissue architecture associated with AD development and progression. Another use case is to apply RESEPT on tumor dataset named glioblastoma, and glioblastoma from 10x Visium website was downloaded and input into RESEPT. We observed the glioblastoma lesion were segmented into segments, while the segment 0 is a background noise, and the true segments are from 1 to 7. To better interpret the biological insight of 7 segments, we collaborate with the 2 experienced brain cancer pathologists from Department of Pathology. We distinguish from morphology of segments we observed segment 3 contain a large number of tumors, segment 6 contain a large number of red blood cells, and segment 7 contain a large number of infiltrating glioblastoma. To support morphological observation, we checked gene signatures of each segment. The glioblastoma classic marker CHI3L1 expression was highly expressed on segment 3, and also geofunctional enrichment pathway also indicate there is some functionalities of red blood cell on segment 6, and the true infiltrating glioblastoma markers were highly expressed on the segment 7. So overall, we confirmed RESEPT has potential clinical and prognostic values on this application. To sum up, we developed a novel deep learning framework named RESEPT for characterizing, visualizing, and interpreting health or pathological tissue architecture. In the future, we may apply point cloud to other non-lattice pattern spatial data because the current method is working on lattice pattern spatial data. And further, considering some colorblind people, we may use a different color system, such same YK. If you have interest on using RESEPT, please find the link address at the link session. And thanks again for your attendance for my poster presentation. I will be happy to answer any questions from you.